This is the finish line. Over 9 episodes we built every piece of a computational drug discovery pipeline. Today we chain them all together into one reproducible mini-campaign against EGFR, starting from raw public data and ending with a short, ranked, toxicity-clean list of candidate molecules, each confirmed by docking. Let's run the whole thing end to end. Here's the full funnel. We start with about 1,000 measured EGFR inhibitors from ChEMBL. We filter them for drug likeness. We fingerprint them and train a QSAR model. We use that model to screen a library of molecules it's never seen, gate the survivors through an ADMET toxicity check, and finally dock the ones that remain into the real protein. 6 stages, each from an earlier episode, now one continuous flow. And here it is in code, the entire series compressed into one script. Load ChEMBL data, filter, fingerprint, train the model, screen the library, apply the ADMET gate, dock the survivors, and keep only the strong binders. Run it, and out comes the campaign result, a handful of candidates, each carrying a machine learning potency, a docking score, a drug likeness value, and a clean toxicity profile. Look at what the pipeline delivered. From 1,000 molecules down to a shortlist that passes 3 independent tests, a learned potency prediction, an ADMET screen, and physics-based docking below -8 kilocalories per mole. The 2 top candidates carry no toxicity alerts and good drug likeness. That's exactly the profile you'd advance to synthesis and a real assay, and every number came out of code you can run yourself. Step back and look at how far you've come. In 10 episodes you went from loading a single molecule to running a complete, end-to-end discovery pipeline, representations, real data, filtering, similarity, machine learning, docking, screening, and optimization. Every tool was free and open source, RDKit, ChEMBL, scikit-learn, smina. And the target was EGFR, but the pipeline is general, pointed at another target's data, and it runs. That's the series. You didn't just watch drug discovery, you coded it, from the first molecule to a confirmed shortlist. Everything is in the public repo, clone it, run the notebooks, and point the pipeline at a target of your own. If this helped, star the repo and subscribe for more hands-on life sciences AI. Thanks for coding along, and I'll see you in the next one.